and decision making and support context-dependent behaviors. And finally, something that we all think about when we think about brains is that you could form memories and learn, but we know that all of this, you don't need neurons for that. But some neurons can also do this, and they have some different kind of properties that could happen. So now the question is, how do we build machines with nervous systems? And you can have two approaches. Well, that's my definition of two different main approaches. One would be this rational engineering approach. And this is, I was very happy. I was not happy with any of the images that AI made. I tried different things, but I was happy with this one. That you would have some neurons, and you kind of like the same way as you wire electronic circuits, you wire up neurons and have them do something. And a really kind of cool, elegant, conceptual example of this are these Brandenburg vehicles. So if you're familiar with these, Valentino Brandenburg has this book called Vehicles, in which he has these machines that he's thought of like very simple, basic machines that have sensors and motors and can perform particular kinds of behavior. And usually, like you would see, okay, if an animal does chemotaxis or phototaxis, it looks complex, but in this book, he's trying to say you can get this very, very simple wiring diagrams of sensors and motors. And the concept here is that what you need to do in order to design these vehicles is to first define the desired behavior. You say, I want to get this behavior out of this machine. This is kind of your rational engineering hat. And then say, okay, what kind of elements do I need to generate this behavior? I need a series of sensors, I need some motors, I need them to be, okay. I think I'm open. Thanks. Yeah, so gather your elements, sensors, motors, some wires to connect them, and then connect these in a logical way that makes sense. So for example, the simplest vehicle number 1 of Brandenburg, do you see this mouse?
Yes.
So this is vehicle number 1, it has a sensor and a motor, and they're just connected directly to one another. And let's say if you have this source of stimulation of the sensor, say it's light, if there's light in the environment, this sensor becomes activated and the motor would move faster, and this is going to just go in a straight line. This kind of makes logical sense. Then you can make this more and more complex. So if you go to vehicle number 2, there are 2 forms of vehicle number 2. 2A has 2 sensors and 2 motors, and then the sensors are connected to the ipsilateral motors. And what the system does is it always goes away from the light. And the way it works is simple. The one that's closer to the light makes this motor move faster than the other one, and then this will go the other way. And if they're connected contralaterally, then this is going to do the opposite. So on paper, this looks very elegant and cool. Can you actually do this in real life with living cells? That's when it becomes a little bit tricky. So here's an example of it's something that is similar to vehicle number 1. This paper is really cool. They tried to build this biohybrid swimmer robot. So again, they're gathering the elements. So they need some neurons that are light sensitive. In this case, they made these neurons light sensitive by genetically modifying and having them express tau-rhodopsin. That is, makes them respond to light. So you can think of these neurons as sensory neurons. Then you have some muscle cells, which they are the motors. In this case, they need some kind of synthetic scaffolds to have these things as the body, have these grow on, and they use this matrix to grow the cells on. And this is supposed to be in a liquid and move around. So this is what they have this kind of guided self-organization, and they build this robot that kind of swims around. This is a little bit more realistic image of what it looks like. It's this kind of design that they have this scaffold design, and then they have a place where neurons grow, a place where muscle cells grow, and naturally these neurons they're motor neurons, so they kind of naturally go towards the muscle cells. So this is the real life image of this swimmer robot, and on the right side you'll see optical stimulation. That's when they start stimulating with light. Then you see an image of muscle contraction, then you see swimmer. So at some point you'll see that it's going to wiggle, and that's when the light is. Yeah, so you can see that it's wiggling. And it's, oh, so pay attention to both the space, like the spatial scale and the time scale. So you saw how it moved from here to a little bit further in about a minute, so, or 20 seconds. So it's moving very, very, very slowly. It's kind of moving, but there are still a lot of challenges in building these kinds of robots. They're not yet fully self-organized. They're not fully biological, so not biodegradable. So if you want an application like that, there are still a lot of mechanical structural challenges with synthetic scaffolds. You still need external stimulation to make them move, so they're not autonomously responding to stimuli in the environment in a way that you want them to. And if you want to make a more complex wiring diagram, kind of like this vehicle number 6 of Brandenburg, this becomes really very, very difficult. If you have different types of sensors, different, positive and negative connections, so this is going to be very, very challenging. So this is one approach. The other approach is to, as Mike gave an amazing talk this morning, and you're familiar with, kind of explore the space of possibilities and just, ask the question, how do groups of cells self-organize in novel contexts and embodiments? And to what extent can cell assemblies adapt to completely new environments? What would they do? On a very short time scale, as Mike mentioned, not an evolutionary time scale, but in a developmental time scale, what can we really get? And so kind of observe and then try to understand what are the capabilities of these living cells and, in the new embodiments. And based on this understanding, try to build machines that basically wire themselves up, so that you don't need to, micromanage. And I really like this image from one of Mike's papers, where you have the space that's in this kind of, it's three-dimensional here, but whatever, n-dimensional space where you have a natural development, you have a rational engineering side, which we talked about, and you have this whole other space that is open for exploration. So that's a space that we're going to dive into. And this is now telling you about what I did with the work. Okay, so a little bit more background. So I'm going to not spend so much time because we're going to have lunch, but this is exactly along that animals that have developed in the context of evolution, they have their nervous system, and their nervous system have evolved in their specific body plans, or vice versa, they kind of evolve together. In the specific behavioral niche and natural environment that they have to grow in and behave in, and kind of, it all relates to that. But what happens if we take neurons from one of these organisms and just put it in a completely new body? And this is what the question that Mike excited about. And in this case, you've seen this before, so this is kind of familiar. You have a motile body that is your xenobot. What happens if you give them some neurons? So the questions are, what happens to their morphology, what happens to their behavior, what kind of connectivity do you get? So this you've seen again before. As you know, xenobots or biobots, I kind of use them interchangeably in my talk, are made out of this top part of the Xenopus embryo, and you've seen this video before by Doug. It's very cool. You see that they move around in a dish, so I'm going to skip this. I've seen this many times. This one, it's a little bit zoomed in image of a xenobot, looking at these multiciliated cells. So these are individual cells with the cilia on them. I like this video just because it shows how beautiful the cilia move. And these are the motors that we're talking about. So these are motor elements. And this is how they move around. So in order to make the neural version of them, so this was what we tried. So there's one interesting fact that is already known, was that if you take this top part of the embryo, Xenopus embryo, and then set it in a dish, it would heal itself and turn into a xenobot. If you do not do that, if you take that same part and then dissociate the cells and put them in a dish and disperse them so they're far from each other, and wait for 3 hours or more, then all these cells would become neurons instead of skin cells, which they normally become if they're together. And the biology of this has been studied before, and it has to do with some signaling, BMP signaling, that if cells are close to each other, they inhibit the neural fate. But if you take the cells apart, they can't have this inhibition on one another, so they become neurons. So that's their default fate, which is normally inhibit. So for the actual making of this neurobots, which is a little bit more painful than making the xenobots, you take a whole bunch of animal caps, or the top part of the embryo, dissociate them, wait, reaggregate them, and then manually, and then cut another animal cap from another embryo, which is kind of timed to have the same age as these, and then put them together. So the first time I tried this, it's like, okay, what's going to happen? Just stick these cells in. Is it going to just spit it all out, or what's going to happen? So it really was cooperating. So there was like everything stuck to each other, and they closed really beautifully. So nothing went, it was perfect. So it closes up similar to a non-neural one heals itself. And then after a few days, also that was another question: is it going to live? Is it not going to live? It lived, and after a few days, it also starts moving around. So this is just the image of that cartoon. So there's like an animal cap, and inside of it are these clumps of pre-neuronal cells, or neural precursors, were implanted, and you can see that after a few days, it's your neuron kind of still surviving and moving around. This is one of my favorite videos of one that is moving around the dish. And this one has very particular, interesting pattern of movement, that it kind of looks like some kind of repetitive spirograph. And yeah, so this is showing that it moves around and is alive and happy. So I'm going to give you a summary of things that were different. There are a lot of things were similar between xenobots and neurobots, but some of the differences were the neurobots generally tended to be more elongated. And interestingly, we found that this level of elongation is correlated with how much neural growth was inside. So the more neural growth they had, the more elongated they became. The other thing is they became larger, so than biobots or xenobots. And one other interesting thing about them was that the density of multiciliated cells on them became smaller. So whereas xenobots have very dense kind of uniform multiciliated cell distribution on them, neurobots have a more sparse, more asymmetrical distribution, which was interesting. I'll show you pictures of that later. Then looking at their behavior and trying to see if there are differences in the way that they move spontaneously. This is an experiment that's kind of set up that we used. We put one neurobot per well in an small 8-well plate. And so you can just watch them for the half-an-hour periods is what I used, and try to quantify the way they move spontaneously and see if there are any differences between them and biobots. And you can appreciate that they all have different individualities. So one of them, and this is, by the way, the same with xenobots. They're all different, move differently too, to some degree. But yeah, it's just amusing to watch them move around in these different ways. And they're pretty consistent. Like you could measure, this guy would just go on and on doing the same thing. But sometimes they do change. So we can track these using a computer program to look at their trajectories. And these are 4 example trajectories. This is from the video that I showed you, is the one of these loopy things. And the color code here is time. I think purple is early and yellow is late. And this was work done by a technician, Lori, who was working with me. She did a lot of these tracking and analysis. And yeah, so these are 4 examples. This is doing this spirograph thing. This was doing like a small circle, and then it changes to a large circle. This is just doing the same circle the whole time, and this is just going around. So in order to kind of quantify more than just describing this, we did this spectral analysis where we looked at more details of the movement by kind of separating the x and y axis in this trajectory and looking at the frequency spectrum to see what kind of shape we get in the frequency domain. So for simple, like circular trajectories, if you just projected an x and y axis, each of them are just going to be a sine wave. And then if you look at the frequency content of a sine wave, you would get a single peak. But for the more complex trajectories, you get a more complex kind of combination of sine waves. That is something like this, and you get multiple peaks in the frequency domain. So simply we took the number of peaks in the frequency domain to kind of come up with a complexity index to see whether or not neurobots have a more complex trajectory on average compared to non-neurobots. And we see that, yes, they're a little bit more complex. They're not hugely, vastly more complex, but slightly more complex trajectories. And this complexity index is not really just a simple correlation, just only related to their roundness or to their area. So there's something more than that going on. We have not yet quantified the relationship between the way the multiciliated cells are distributed and this complexity, which is very, very important. That could be a reason why we're getting more complex trajectories, because they're not as uniformly distributed. The other reason could be that because of the neural activity, you might be modulating the moving of the cilia, you could be doing anything like that. So we haven't yet completely discovered what is coming from where, but this could be a hypothesis. So, as promised, this is a picture of a stained neurobot with acetylated alpha-tubulin antibody that labels multiciliated cells but also neurons. The color code is just showing, it's just like a Z-projected confocal image, it's just showing the depth. And as you can see here, you have this kind of longish neurobots, and you see that a bunch of multiciliated cells are on one end of it, and then the other end has less, so it's kind of not uniformly distributed, and the shape is also very interesting curvature. If you look inside of it, then you can actually see the neural processes that are growing in there. It's a lot of things that are happening, so there are neurons trying to find their way, see what they're supposed to do. And then you'll see very interesting things, such as this one, that some processes, sometimes you see this going on a straight line as if like they know where they're going. And yeah, so this is very interesting. And then we see also some of the processes going to the surface. So this is interesting because if you want to change anything about the movement by neural activity, you need to go there somehow to communicate with the surface cell. So the fact that we see that they do go there tells us that at least the physical thing is there and could have the ability to modulate motors. This is the same bot, but just focusing on one part of it and looking at, again, the acetylated alpha-tubulin stain plus a nuclear stain. The nuclear stain is in blue. And you'll see here is these clump of nuclei. This is the bunch of cells that were implanted, the precursor cells. And you kind of can go in and look at these very thin neural processes that's here. I'm just wanting to show that they do go to the surface of the bot and kind of go close to where multiciliated cells are. And one of the million future plans that we have is to quantify this in a lot more detail to see if there's actually synaptic connectivity, to test functional relationship between the movement of the individual multiciliated cells and the neural activity. So, but at least anatomically, it looks like they're going. This is another example of another bot showing the neural processes that are going towards this. And you see this in almost everyone. And again, this is the clump of cells in the middle. The other thing is that if you look at all these different neurobots, they all seem to have some elements, some properties that are similar, but some that are very different. So similarity is a lot of them are more elongated. The other similarity is that a lot of them have areas where you have less of the multiciliated cells and some more. And it looks like those areas are kind of around the time when there are neurons, around the locations when you have these neural clumps. So this is one of those questions that I'm really interested in to dig deeper into to see what's the relationship between neurons being in an area. Are they suppressing expression of cilia, and what is the relationship they share? This element, the microtubules that are inside them. So this is why they stain together. So there must be some relationship between gene expression. That would be very cool to investigate. I'm just showing you various examples of these. Another thing that is common, well, they all emanate from that nucleus, but that's expected because that's where I put them. So future work, you can look at various configurations, not put these clumps, just put individual cells. But in all of them, the other thing that we see is that there's this kind of empty-looking space that you're still super puzzled with. So there are these nuclei, but then around them there's this seemingly empty space. But at the same time, it can't really be empty because you see these straight going processes of neurons. And we have various hypotheses as to what this empty space could be. And we think that at least some of it may be filled with something like extracellular matrix. And we're digging deep into that, and we're planning, hopefully, experiments soon where we're going to do spatial or transcriptomic style, looking at where things are. But some images, I spent some time taking some EM images of these, and some of these EM images indicate that there might be something like collagen fibers in some of these empty spaces. This is kind of an EM, this is an EM image of a neurobot. There was a lot of things that you don't know what they are in these images, so that's very hard to read. But this is part of that empty kind of space, that kind of, these are cells around this area. And then in the middle there is a whole series of objects. Some of them might be axons, we have to look into that. But if you zoom into this region, you see these beautiful kind of frosty looking structures that they might be collagen fibers. So these are some of the hypotheses that we're going to test. Okay, the neurons inside the neurobots are active. So just to test whether or not they're all alive or dead and what they're doing, I made some neurobots, implanted them with neurons that express GCAMP. I think it was either Doug or Pi that injected those embryos, thank you, with this GCAMP construct. So then I implanted them in the neurobots and just looked at them using this custom microscope. And this is what I was talking to Stephen Wolfram yesterday too, about the challenges of doing these kind of imaging. I think with the neurobots is even more challenging because the neurons are on the inside, as opposed to doing wide field imaging, you best see the outside cells. So it's hard to get enough number of them, but you can still, correct for the motion and then look and see that you can see blobs that are firing, coming on and off, so that the neurons are active in there. And you can define some regions of interest, look at correlations. So these are experiments that hopefully you can do more of in future to look into more detail of like how functionally connected they might be, as Thomas was describing yesterday. But they became very challenging. Our initial goal was to start to see if there's any relationship between neural activity and movement. And that's particularly challenging because you need to do long periods of recording and you need to keep the same neuron in focus the whole time. And these are hopefully soon in future we can overcome these challenges. I don't think they're impossible, but we need more work to make them happen. Another kind of indirect measure of whether or not neural activity might have an effect on the movement is we treated these with pharmacologically using this seizure-inducing drug PTZ, which is normally used to study epilepsy in animal models and seizures. So we wanted to see, okay, if there are neurons in here and this PTZ drug is a blocker of GABA, normally increases excitability, do we see that neurobots become more active? We dump PTZ on. And as a control, we wanted to compare to see non-neurobots, like xenobots, what happens to them. So to our surprise, what we found was that if you look at that complexity index that I introduced earlier, and see how that changes with adding PTZ, and compared to pre-treatment, every circle is an individual bot. So you'll see that with biobots or xenobots, most of them reduce their movement when you put PTZ on them, which is kind of interesting. But with neurobots, it was kind of, they could go either way, which was also very interesting. So, and this was, if you looked at compared on average neurobots and biobots, there was a significant difference of PTZ effect. But that was mainly coming from biobots going down. So this was interesting. So there's a difference. There's an effect on the non-neural bots as well, which tells us that cells on the surface of the biobot might have some kind of GABA receptors that respond to PTZ. Maybe there are some papers that shows these mucus secreting cells have some GABA receptors. So maybe there are changes in release of mucus that makes them move differently. But whatever effect it has here, we don't have it here, and we're kind of going the other way too. So this means that because the body of neurobots is the biobot, this means that whatever they're doing, they're actually overcoming that inhibition that's just on the biobot. So it is kind of consistent with this idea that it is excitatory on neurobots compared to biobots, but we have to explore it. Right, so another thing that we looked at, we wanted to see the pattern of gene expression in neurobots and compare them to non-neural bots and to shams. And shams are ones that are made the same way as neurobots, but when I implanted this clump of cells, didn't wait 3 hours for them to become neurons. So just immediately after this person, then we aggregated them and implanted them inside. And we put multiple of these bots inside Eppendorf tubes and for RNA sequencing, and bulk RNA sequencing, and see how they compare to one another. And here is just like a general correlation matrix that if you see this large box here, these, so on the X and Y axis, we have the 4 neurobots samples MB1 to 4, then we have the 4 shams and the 4 biobots. You see that shams and biobots were highly correlated with one another. So even though this mechanical kind of implantation effect did not seem to hugely impact gene expression, so they're still very similar to biobots, and these two groups are very similar. But neurobots are very different from the other ones. So the effect in the change in gene expression should be coming a lot from the fact that they're neurons. So this is another way of showing this, just pairwise comparison of neurobots versus biobots and shams versus biobots. The volcano plot is showing, these are all the genes that were found, and then looking at log fold change and how significant the change was. Everything that's shown in red was significantly different. And as you see, shams and biobots had only a few. There are some that were different, but there are very few relative to neurobots to either. And there was a lot of genes that were upregulated in neurobots compared to xenobots. And then we can see what these genes are. So this is showing you kind of pathway items of the genes that were upregulated, but they were upregulated more than 4-fold. So there are a lot of genes, so we chose 4-fold upregulation. And so I'm just zooming into the top 10 of them. And as expected, the first one is the nervous system development. So you expect that you're having these neurons. There are also categories of synapse and transsynaptic signaling, which means that there are synapses being formed, and so the genes are there. It would be interesting to do, quantify where exactly these synapses happen. Do they happen between neurons and other cells? So normally, in normal Xenopus embryos, neurons don't innervate multicellular cells on the skin. It would be really cool to see if they do in neurons. And very, very surprisingly, we found that there is this category of visual perception. There are a lot of genes that were upregulated that are involved in building the lens, building the eye, building the retina, building retinal ganglion cells. So this is very, very interesting. And yeah, basically all the genes that are you need for building eye and retina are there, like rods and cones, retinal ganglion cells, bipolar cells, amacrine cell genes. All of these are present. And the other thing, the previous work, Megan Sperry really helped a lot with analyzing these RNA-seq data. She's another staff scientist at the WCDO. And then Leo, postdoc in Mike's lab, helped with another series of analysis looking at gene network analysis and protein-protein interaction and seeing how densely connected these genes that were upregulated are. And the more densely connected these networks are, it means that the genes that are upregulated, they're just not isolated random genes that are coming up just randomly. It means that it's a whole program of a pathway that everything is coming in together. So all these visual processing genes, it's not just a raw gene, it's like the eye gene, the retina, the lens, the cones, the retinal ganglion cells, so the whole thing is coming up. So this was very interesting to see that this whole program gets recruited. And that's also the same for various other pathways, like neurotransmitter receptor pathways, there are a lot of densely connected upregulated genes. Yeah, so I hope I didn't take too long. I'm stopping people from lunch, but future directions are, the list is infinitely long. But the next steps is to somehow standardize generation of neurobots. So right now I've been implanting them manually and just see what happens, and that was great. I think it was a good start to see what are the range of things that we can get with this method. But in future, I want to work on various ways of implanting, counting the number of cells, maybe inducing certain neuron types, playing with various parameters, and then quantifying architecture, spatial organization of the cells that develop in the end. Assess presence of neuromotor control through optogenetic and pharmacological stimulation of neurons and measuring the behavioral output. And also investigate presence of sensory modalities that neurobots respond to. So the first one is light. So I have to design experiments to see if they really reliably respond to light stimuli and what kind of behaviors can them. And then longer term future directions is to build self-organizing neurobots with predictable sensory motor behaviors. So this is, I've replaced the Breitenberg vehicles with these. So maybe you can build neurobots that wire themselves up, that go away or towards the light. So I think this is a really cool thing that I'm excited about trying, is to see if we can basically, as they are developing, if we provide the right kind of environment for them so that they would wire themselves up in a way that they behave this way. So the environmental factors, I think, are very important. That's kind of the same thing that during evolution, when we were talking about animals are kind of adapting to their behavioral niche. So if you present a behavioral context in the course of development, would they develop behavior? Ones that respond to multisensory stimuli, for example, go towards light, go away from chemical, and maybe those that can learn from their past experience. And with that, I would like to thank people at the Wyss Institute and Mike's lab and Mike, and also various sources of funding. And thank you for your attention.
Thank you, Haleh. Plenty of time for questions, please.
I have a quick question. Yeah. Actually, two questions. One is in terms of the amount of cells that essentially are in the mature neurobots. Are they similar to what's the number? Also about 1,000 or something like that, or how?
1,000. So there are definitely more number of cells.
More number of cells.
Yeah,
than,
because
than
you have
biobots.
the xenobots plus the neurons that are inside there.
Right.
Yeah. I don't know exactly the number. We have to count them, but.
But do those cells actually, once you put them together, they divide, right? So it grows. And the second question, because the elongation, it's very speculative, but do you think it's trying to grow a spine?
Like an actual vertebrate, like a spine?
No, not a spine in the sense of just like linear.
Good question. I don't know. Yeah, good question. Maybe if there are markers of like spine formation, maybe we could look at those and see if there's
And what's the last question? In terms of the proportion of neurons to somatic cells, is there a big variation, or it's all a pretty stable ratio as they develop?
Very good question. Yeah, you're saying in the course of development, if the ratio remains constant or one speeds up compared to the other.
Of what you have observed in the real things, do we know?
No.
In the sense that, is there more of one to the other, or is...
in general, in the beginning, like if there's more neurons than the outer cells, or if there's... That's a good question. I have a hunch that there's, I don't know, probably more outer cells than neurons, but I haven't really quantified that.
So the expression of the visual genes is really interesting, and I'm thinking about the neuroorganoid results, where they kind of form these dark patches that turn out to be kind of proto-retinas. Have you looked at whether there's any similarity there? Is it possible that the neurobots are behaving like the actual neural organoids?
What do you mean by neural?
So you see it's like an organoid that's just neural tissue
Okay.
allowed to develop as itself. It's not wrapped in, somatic xenobot tissue. And they form these really weird, like, dark blobs on the surface, and no one knew what they were until they sequenced them and discovered that it's proto-eyes.
Interesting.
And that freaked everybody out. They're like, oh god, they're growing eyes.
Yeah.
We could talk later about
Yeah.
that. That could be an interesting thing.
I could definitely. I mean, there are genes, eye-related, lens-related, so it's possible.
Quick question. That was a great talk. I was wondering, in terms of the cilia distribution, you mentioned that it becomes asymmetric, right?
Yeah.
Is it asymmetric in a consistent manner, or is it randomly asymmetric when you make like different neurobots?
Yeah, that's a really good question, and I think at some point I was discussing with someone who would want to quantify exactly the distribution. There's a lot of them that have this shape, but they're not all like that. Like this pattern of like this, things on one side, a lot of cilia, and the other side not. So there are quite a few that look like this.
But do all of them have the same cilia distribution?
What's that?
Do all of them have the same, like all of them that look like that, the elongated one? Do all of them have that exact similar kind of asymmetric distribution?
I would say so. I have to really go back and look, but when I was going back and kind of collecting these series, like this one is a little bit different. This is not as elongated and it's more round, but it's still like they're all kind of pushed to one side.
Because that would be a great mechanism to actually predictably do a asymmetric cilia distribution to gain control over how they move, right?
Yeah, definitely.
Super cool.
Yeah.
Wonderful talk, thank you. I have a question on the mystery of the empty space.
Yeah.
Have you checked whether there is hyaluronic acid inside? Because it's a common hallmark of cardiogenesis and neurogenesis, what we call the cardiac jelly. Without that, even the neural development doesn't go through. You may simply use 4-methylobelliferone, 4-MU, which is a specific inhibitor of type 2 hyaluron synthase. If the whole stuff doesn't go through, that means that it's a fantastic replica of a mechanism for cellular polarity at the chemical electrical level.
Wow. Yeah, I have to write down the stuff
you can use this. It's a molecule that specifically targets type 2 hyaluron synthase. Maybe a hypothesis.
Yeah.
Yeah, thank you for this experiment, because I've been wondering about this for a while. What's going to happen if you have a neuron there? So one question here is, have you noticed the significant difference in terms of memory between the neurobot versus the biobot? Is there any way to quantify that?
It's a very good question, but I feel like we need to design really specific behavioral tasks to look at that.
One thing you could look at is like the autocorrelation function of the trajectory. Does it tend to return to certain patterns more or less likely than a basal xenobot? That should be mathematically pretty easy to define.
Yeah. With that, it'd be good to find out exactly between these two. What is the memory holding difference?
Yeah, that would be very interesting. I
That'd
think
be really
it'll be a bit tricky because, like, even a simple circle that a lot of xenobots do is always coming back, but then this more complex patterns may not go back to the same place. But we kind of, I'm happy to discuss more how we can disentangle this, like either doing something really simple, still is considered having more memory than like something more complex where.
Let's talk about that over lunch, because I think I have a response. Yeah.
You might have mentioned this, sorry if I missed it. The big problem with the stuff is
Can you?
Yeah, sorry. If you said this already, sorry. The big problem with organoids is the necrotic cores. Do you ever see evidence of this? And if not, do you think they're overcoming it, or is it just that they're so much smaller, or what's?
Yeah, that's a good question, and I guess we could look into markers of like cell death. I don't know if there are like antibodies or some kind of staining that we can use to see is the inside full dead tissue or what.
I
But
think in organoids, they just, after a while, I mean, at the end point, they just kind of slice them down the middle and do like a live hits.
yeah, actually we were gonna slice a whole bunch of them, so. But I
That'd
don't
be
know.
cool, because if
after,
they somehow overcome the necrotic core issue, whatever you're doing, organoid people need to do, because like, basically kill them.
Yeah, they definitely live at least as long as the non-neural ones. So
Okay, cool.
I don't think that they're not dying earlier.
So the lifespans are
Pretty similar.
pretty similar, yeah?
Non-serious question. Do you think that Planaria would like to eat neurobots?
I was thinking about that.
What if we had neurobots, right?
Because like there's different types of cells. Do you think it would be tastier for the Planaria?
She just wants lunch.
I might be hungry.
If your mind was blown by this talk and you need someone to smush your mind back into your head, well, obviously we've got an expert here. Let's thank Haleh again. Okay, we hope you enjoyed this morning's session. Before you start mobilizing, a couple of notes. We're going to be leaving this room and not coming back. So, Pi, is there any last minute things you want to tell us about the Fight Club taking place?
I was looking at it while watching Haleh's. So it looks like that both the sides are winning in some ways, because I see only 5 Xenobots in the Planaria sphere, and we started with 10. So
They making Planaria. I think Xenobots ate them.
as I said, they're camera shy, so they're probably sticking to the sides. But yeah. So it looks like they can eat them, but they don't like them as much as the Xenopus embryos. So it's different, but yeah. So I guess both sides win. I will have to look at the other video because it's too small for me to see from here. The Paramecium one. I'll probably peer at it maybe over lunch or sometime and maybe let you guys know what I think.
Neurobots. They might be tastier.
I know, right?
Okay, again, before we mobilize, we're losing this room, so we're going to leave and not come back. We are still in academic mode, which means we have to clean up after ourselves. There's another group coming in, so please make sure to take everything with you. Everything that's on the table, any garbage, pieces of paper, just take them and leave them on the back table. We are going to be going back for lunch to the same room we were in before. You leave this room, keep this room to your back, leave the building. The pretty building that's on your left, go into that building, 3rd floor, John Dewey Lounge. Leave this building on your left, 3rd floor, John Dewey Lounge, and we will see you all there for lunch. Thank you. Sorry, I know we were losing this room. Did you get any instructions from Catherine about the microscopes? Like, are you planning to?
No, I'm going to take them. I'm going to wrap up everything.
Everything now?
Yeah.
Okay, so my people should stay. Cam,
No. I think Krishna and I already have. So we have a card.
Sure?
Yeah. So
We don't need that stuff?
no, I don't think so. We loaded it out. It will probably take us 15-20 minutes, but yeah, that should be.
Alright, have you message us?
Yeah, I will message you or message you. No worries at all. So the recorded video on there, which I guess is still going on, how can I get it? Yes, I do have it.